Hello, senior scholars. Welcome to your brief introductory video to Unit 7, Topic 6, which is studying pathogens. I will not be doing this full topic. I will simply be doing the notes that will allow us to speed through some things. So first of all, let's start with some review. The difference between a pathogen and a non-pathogen. Pathogens cause disease in the organisms they infect. So they cause disease. Therefore, when we use our basic understanding of how words work, non-pathogen doesn't cause disease. But there is an exception to this. We are assuming that the people we're talking about here, or the organisms we're talking about here, are normal, immunocompetent individuals. So, if someone is immunosuppressed for any number of reasons, maybe they're receiving chemotherapy, maybe they have an autoimmune disorder, maybe they have HIV or AIDS, there could be a lot of reasons, but a non-pathogen, meaning something that is not dangerous to the majority of people, could still infect and cause disease in an immunocompromised individual. Now, a great example of this is the tables you work on every day, which I'm currently showing. This is clearly not a microscope, but this surface covered in bacteria. So if I were to have a paper cut and then put my hand on this surface, it would actually only become infected if the bacteria on the surface were pathogenic, because I am immunocompetent. If I was immunosuppressed, I might be able to be made sick by anything, but as an immunocompetent person, even if the bacteria on the surface of this table made it into my paper cut, my immune system would fight them off before they had an opportunity to infect me. So those are our two key differences, and to be clear, these are things you learned last year, and these are going to be core terms moving forward for the rest of time, we're going to say. Now, I would encourage you to pause the video and then answer these two review questions that pull on what you know about BSL levels as related to these words. So I would pause the video now and answer these two review questions. You're back? Great! That means you've answered these two review questions. If you haven't answered the two review questions, pause the video now and go back and answer them. Now, I will not be filling in the bacteria column because that is review. I want you to, after we fill in the other two, pause the video and fill in the bacteria column because, as I said, it is review. But I am going to go through the types of pathogens, specifically viruses and prions. So there are three categories of pathogens that are the most common. There are, to be clear, some infectious protists, like the paramecium that causes malaria, but those are quite rare. These are the three most common forms of pathogens that can infect other living organisms. The first one is bacteria. The second one are viruses. The third one, these are probably brand new to you unless you've done some very high-level biology, prions. So, what are viruses made of? They're made of two things and two things only. They are made of a protein coat. Now visualize that word. Tick, tick, tick. What do you think a coat would be found? Like a coating? Not like a coat you wear in the winter, although technically it's called a coat because it goes on the outside. Yes, that's right. The protein coat goes on the outside. And then inside that protein coat, you will find genetic code. Yes. Now, viruses are relatively special in that their genetic code could be DNA, just like cells are, or unlike cells, their genetic code could actually be RNA, meaning they don't have any DNA, they just use RNA. I'm not going to answer the question for you right now of are they alive, because you should be able to answer that question already based off of topic 2, cell theory. So I'm just going to let you do that. Now, how are they classified? I'm going to go into that in the next subtopic of these notes, but they are basically classified by their shape of their protein coat and whether they use RNA or DNA. Examples of viruses include HIV, the human immunodeficiency virus. Obviously, COVID is an example of a virus, and any kinds of influenza, more commonly known as the flu. The common cold is also caused by viral infections. To be clear, literally thousands of influenza and common cold viruses exist. Common cold virus is actually known as the rhinovirus, but there are thousands of versions of them. But all of these guys are viruses. So, moving on to prions. Prions are made up of actually just a protein. That's it. They don't have any genetic code. But here's what's crazy. They act like a virus, meaning when they infect you, they actually forcibly refold the host's proteins to be like they are. In other words, the prion takes other proteins, refolds them to become other prions. So it's essentially an infectious protein. That's pretty crazy. We actually only discovered prions in the 1970s, and we didn't really do much with them until the 1990s. Now, I'm going to leave Is It Alive for class, but you should have an answer decided. And how are they classified? This is really in A. Prions are a high enough level concept that if there are different classifications of prions, you don't have to know them. But examples of prions include mad cow disease, which in humans is known as Creutzfeldt, that's a Z, Jacob disease. Same disease, but in humans, in cows. What this does is if you get Creutzfeldt-Jakob disease, first of all, you're going to die, but you're going to die in about 30 years. And how you're going to die? The prion over those 30 years is slowly but surely going to refold all of the proteins in your nervous system to become other prions. So because it's growing exponentially, not only will the disease kill you, the disease will get worse and faster as it goes on. And then you will die an incredibly painful death because your brain and your nervous system have been turned into Swiss cheese. You will also develop extreme dementia in that time. It is a terrible way to go, and it is why I was a vegetarian when I traveled through Europe in the early 2000s. All right, if you have not already done so, pause the video and fill in the bacteria. My phone is telling me I'm low on storage. Let's see if I can finish this without having to go to a second video. All right, the classes of virus are 3. We have a bacteriophage, a retrovirus, and a DNA-based virus. Bacteriophages only infect bacteria. That's right. This virus is a non-pathogen to us, but a pathogen to bacteria. Now, how can you tell a bacteriophage? They have a crazy shape that looks like they were designed by a kindergartner who was told, draw what you think an alien virus would look like. So they have this kind of diamond-shaped thing. This is the protein coat. Inside the protein coat, they either have their RNA or their DNA. But then they have these other proteins that look like these spider legs coming out of them. And they use those spider legs to grab on to the bacteria that they infect. So if you were to take those off, they would not be able to infect the bacteria, but don't those just make them look super creepy? Yes, yes it does. Then we move on to retroviruses, which are, there's a memory device here, R for retro, you should remember is R for RNA, because a retrovirus is just any virus that infects eukaryotes. It cannot infect prokaryotes and uses RNA, not DNA, as its genetic code. This is why reverse transcription occurs in the real world, which we use when we're doing genetic engineering. It's due to these retroviruses. Now, a retrovirus is going to have a protein coat. That protein coat could have a whole bunch of different shapes to it, but inside it's definitely RNA, so I've drawn it as a single strand. And most retroviruses have these proteins that jut out of them, like I'm drawing now, which are also used to attach to the cells that they're going to infect. Now, you probably recognize this because the classic example right now of retroviruses are COVID, but please keep in mind that HIV is also a retrovirus. Now, the third category are DNA-based viruses. These also infect eukaryotes, but they use DNA as their genetic code. That means they don't contain reverse transcriptase because they don't need it, because they just insert their DNA into us when they infect us, the dirty, dirty little DNA-based viruses. So they actually have a lot of different shapes. You are far less likely to be assessed on those just because they are super highly variable, whereas these two bacteriophages and retroviruses have a lot of applications in biotech where we've taken parts or bits of them and used them to create techniques or products. So let's talk about, though, a little bit of background about how viruses work in nature and then how we have used that in biotech. So this is viral reproduction, and viral reproduction is known as the lytic cycle. Now, hopefully you remember lytic is from the word lysis, and that means to split. We are going to figure out why we call viral reproduction the split cycle or the lytic cycle when we get to the end. Let us start with step 1. In step 1, now this is a bacteriophage, so therefore we know that this is a bacteria it's attaching to because bacteriophages only attach to bacteria. So here we see we've got our little nucleoid of DNA that is not inside a nucleus because this is a prokaryote, so it doesn't have them. That nucleoid of DNA is just floating in the cytoplasm. You can see the bacteriophage attaches to the cell wall of the bacteria because all cell walls have bacteria. Step 1 is complete. We then move to step 2. If you look at this quite closely, you can